events. One is this sort of PDF here, which is sort of a PDF of the PowerPoint, which is an abbreviated version. And you'll notice that there's also a study guide that sort of goes along with you fill in the blank that contains way, way more information, all right? And we're trying to sort of limit it, so sort of focus on what we're sort of doing in class here today. The pictures that sort of go up here, the pictures you're gonna see here, this is the one that I want here. Yeah, this is the essential. Are often going to contain a lot of information that I need to sort of go over with you here. So we start with the following, is that one of the hallmarks of all living organisms is the need for useful energy. Different types of organisms gain their energy in different ways. We gain almost all of our energy from chemical bond energy from the foods that we eat. Now, when we talk about chemical bond energy, it's probably a good idea to start with the fact that when we talk about the energy components in foods, it's about the electrons, because electrons are the portion of an atom that can vary tremendously in the amount of energy they store. You can have low energy electrons, you can have high energy, you can have higher. See how that works, people? Low, high, higher, even highest here. So when we talk about energy, for example, when we talk about energy in a compound like glucose, C6, H, 12, O, 6. Yeah, I'll do it all myself. There's my glucose molecule here. The energy, almost all the energy in this molecule is going to be stored in the 12 electrons that accompany the hydrogen. When you think of glucose, when you think of a sugar, you should think of it as a 12-pack. A 12-pack of energized electrons that accompany which element? Hydrogen. I'll do it all myself if I need to. Hydrogen, because H stands for hydrogen. You're killing me here, people. I know it's early in the week. You're killing me. Now, how do we get this energized compound to begin with? Well, that would actually go back to photosynthesis, which is something you might have done in a Bio 106 class. Because in photosynthesis, plants will basically take water and carbon dioxide as raw materials, and they strip the hydrogen, they take the electrons, they energize it from the energy in sunlight, and then they store these energized hydrogens on a frame that was made out of carbon dioxide. Now, is it going to take some energy to build the frame?
Yes.
Yes. So just breaking the frame apart, will that liberate some energy?
Yes.
Yes. And so when we talk about the way that we get energy out of it, later we'll talk about sort of two basic mechanisms. One, they talk about substrate level phosphorylation, which means that you just do it by breaking bonds apart, energy is going to be released. You don't need oxygen, you don't need mitochondria. It happens all over the place, a lot of different enzymes. But most of the energy we will see is a process called oxidative phosphorylation, and that's what mitochondria, remember your old friend mitochondria from your 109 days? Mm-hmm. That's what mitochondria are required for. Because oxygen is going to be needed as the final electron acceptor. We're going to take these high energy electrons, bounce them from carrier to carrier, and use their energy to pump hydrogens into an enclosed space. And we allow the hydrogens to flow out. We will regenerate ATP. Don't worry about writing this down. We will see the concepts behind this a little bit later here. So we need to first talk about some definitions of energy. We've talked already in 109 probably about potential versus kinetic energy, correct? Mm-hmm. Potential energy is stored energy, where kinetic energy is energy in motion that's sort of being used. You can transform energy from one form to another, from a form of kinetic into, or potential into kinetic. in every energy transformation, you remember that some useful energy is lost as heat energy. Now, the total amount of energy is going to stay the same. If you start with 100 calories worth of energy and undergoes 17 transformations, how much energy will be at the end of it? 100. You start with 100. I just made that shit up about 17 to see if I could like mess with people's mind, but if you start with 100, undergoes transformations, you're going to end up with 100. If you started with 100 of useful energy and undergoes 17 transformations, you're not going to have 100 units of useful energy left. You might have 10 useful and the rest is going to be liberated as heat. Heat is considered a waste energy because it's almost impossible to transform it into another form again here. So when you take a look at these bonds here and then the carbs and the proteins and lipids, the chemical bonds between them all have some potential energy stored in those electrons here. And it's liberated in these intracellular metabolic pathways, hopefully to form ATP, will then be used to power other reactions. So when nutrients are abundant, we tend to store them in different ways here. In our little 4-step process here, which is a really interesting one here, there's some important terms here. When you say energy is released from exergonic catabolic reactions, there's a lot in that statement. Did you talk about what exergonic versus endergonic means back in your 109 days? You remember this at all? I'm seeing kind of furrowed eyebrows and a little deer in the headlights.
Can we go over?
Look here. All right, so let's talk about what these terms mean. Every chemical reaction requires an input of energy to get started. They call that activation energy. So every chemical reaction requires an input of energy to get them started. We call that activation energy. What happens next will determine whether that reaction is going to be exergonic or endergonic. In an endergonic reaction, all right, the endergonic reactions, the products have less energy. All right, I'm sorry, it takes less energy than it took to get it started. They release less energy than it takes to get started. Exergonic reactions release more energy than they take to get started. Exergonic reactions release what? More than they take to get started. So for example, once you start an exergonic reaction, they tend to be self-sustaining because they keep adding activation energy to the molecules next to them. Burning paper is a great example of something that would be exergonic. Because do you need to add activation energy to get your paper to begin to burn?
No.
Everybody agree? Is this paper like burning right now?
Oh, no.
Am I going to have to add activation energy to get that started? Yes. By lighting it on fire. Once you add activation energy, see how I keep taking your test away from you? Once you add activation energy to the corner of it and get started, well, then it releases what? More energy. And that's going to activate the molecules next to it, and the paper continues to burn. Once you get it started, they tend to be self-propagating. Endergonic reactions release less energy than they take to get started. All right? Which means if you want to keep them going, you have to keep adding energy all the time because the products that they produce contain more potential energy than the reactants do. Photosynthesis is a good example of endergonic reactions. Cellular metabolism that we're talking about here, all right, is a great example of exergonic reactions. When you're building proteins and you're building molecules, they also tend to be endergonic. They take an input of energy. So in living systems, the common way that we provide energy for endergonic reactions is to couple them with exergonic reactions. So a good rule of thumb is that exergonic reactions will provide energy for endergonic ones. Exergonic reactions will do what?
Provides energy.
Provide energy for the endergonic ones. Now, we're not even finished reading the first sentence yet, because that first sentence here says, energy is released. Gesundheit. Energy released from exergonic catabolic reactions. We also need to define what catabolic and its opposite anabolic mean. So catabolic occurs when we are breaking things down. So for example, when I break down a glucose molecule into its components, all right, to liberate energy, all right, that's a form of a catabolic reaction because we're doing what? Breaking things down. The catabolic part means that we are breaking things down, all right? The exergonic portion of the term means that in breaking things down, they're going to be doing what? Releasing energy. More energy than they took to get started. The opposite of catabolic is anabolic, or anabolism refers to what? Building things up. Quick question. Generally speaking, who have larger muscle masses, men or women?
Men.
And why is that? Is there potentially a hormone? It's all about what? Testosterone. Because testosterone helps them to build up more muscle, all right? Would building muscle be considered anabolic or catabolic?
Anabolic.
Everybody agree? It would be anabolic?
Yes.
I'm hoping everybody would agree with that, right? And of course, some athletes have been known to try to cheat, if you will, all right, by, especially in sports and events that require more muscle mass and more strength, by taking drugs that basically mimic the actions of testosterone. Basically, so when they talk about an athlete that's taking steroids, they'll often talk not just about steroids, they use what term in front of it? Anabolic. Have people familiar with that? They say so-and-so has been taking anabolic steroids. Why would they say anabolic? Because anabolic refers to the fact that these are steroid hormones that are doing what? Building things up. That's important because you have other steroid-based hormones like cortisol, which are steroid hormones that are there to, well, have an impact on metabolism during stressful periods and things like this, all right? You already know probably that steroids like cortisone, for example, can suppress immune system, suppress inflammation, but they're not anabolic in nature, are they? No. They're not building up things. So when they talk about somebody who's on anabolic steroids, they're talking about something that's taking a synthetic version of, really a synthetic version of testosterone. It'll have some of those same effects. So when they talk about energy being released from exergonic reactions, all right? Exergonic catabolic reactions means that we are breaking things down. Sugars, carbohydrates, breaking them down here. And then that energy can be used to fuel the endergonic anabolic reactions of ATP synthesis. All right? It's endergonic because it does what? Requires a net input of energy. You got to keep it in energy. And it's considered anabolic because we are building things up. We're we're taking a molecule ADP with just 2 phosphates, all right, adding a phosphate to it that doesn't want to get attached. See this fact that it's negative here? Phosphates have negative charge, right?
Yeah.
Yes, they do, right? And what do similar charges do to each other? Similar charges do what? Repel each other. So because these phosphate groups are doing what to each other? Repelling, it's going to take a lot of energy to overcome that repulsion. I can just hook them together. And then there'll be chemical bond energy stored in the bonds between them. Now, then, I can then use that later. All right, ATP is going to be broken down in exergonic reactions, all right, to fuel other endergonic reactions like the synthesis of protein, for example, or the building of cell membranes, the moving of muscle, or anything like this. So you'll notice in something like this, we're pairing exergonics with endergonic that drives the next exergonic that's going to be used for the next endergonic. You had a question there?
Is exergonic always catabolic and is endergonic always anabolic?
For our purposes, yeah. There are rare exceptions, but forget those. No, seriously, forget those. Now, there is a whole lot of crap in this one picture here. Whole lot of good stuff. But let me just try to summarize what's sort of going on here. Do you recognize this particular organelle here? This is a mitochondria here. And so here's what's happening in this, what's showing here. There are different ways that we can sort of get energy from foods that we consume here. So I'm going to start with this, a simple one here is the process of breaking down glucose. All right. The process of extracting energy from glucose initially is called glycolysis. And the process of glycolysis occurs in the cytoplasm. You don't need mitochondria for the process of glycolysis. Where can I write that? Now, it's going to be useful to keep track of, especially to keep track of carbon numbers and hydrogen numbers can be really very, very helpful. Oxygens can come and go a little bit. I'm not so concerned about that, but keeping track of carbons and hydrogen, very, very helpful. So when I break down my glucose by glycolysis, process of glycolysis, I take with me my C6, H12O6, my glucose. I'm going to break it down ultimately into 2 3-carbon sugars. They're really modified sugars that are called pyruvate. Now, when we were forming this sugar by the process of photosynthesis, I mentioned, is it going to take some energy to actually build the frame? Yes. So we'll break the frame apart, release some energy? Yes. And in fact, we're going to get 2 net ATPs regenerated, just what's called substrate level, just happening in the cytoplasm. Don't need mitochondria for this at all. You get 2. Now in this case, what I'm giving you is the lie, because in many cases, it's just so much easier to lie than to tell the truth. Because sometimes you have to tell the truth, you have to go in the hole, and nobody believes you anyway. So sometimes a short, simple lie is just the easiest way to go. And that's what I'm giving you here, is a short, easy lie. If they ever come back to you, they want the truth. The truth is, this process requires an input of 2 ATPs, and you get an output of 4 ATPs. So if you ever get that to you in the future, you actually have to start glycolysis by wasting 2 ATPs. They call it priming the pump. But then after you break it into the 2 pieces, you get 4 ATPs out of it. So the net gain is still 2 ATPs. Now, back to photosynthesis again. We were energizing these hydrogens, right? The sunlight energy, the electron, right? And we were storing them on a frame that was made out of what? Carbon dioxide. The frame was made out of what?
Carbon dioxide.
And the reason we need to build the frame was to hold on to what? Hydrogens. We built the frame out of carbon and oxygen so we could hold on to what? Hydrogens. Which means that anytime I'm breaking the frame apart, what do you have to account for? The hydrogens. So anytime we're breaking the frame apart, breaking this apart, all right, I don't want to say hydrogens are going to be falling off of it. It's not exactly like they're falling off of it. But we need to remove the right number of hydrogens. Now, these hydrogens are still going to have lots of potential energy in them. All right, because the electrons that they accompany are energized electrons. That was way back in photosynthesis again. So we have a taxi. We have a couple of different taxis, but the most important taxi we have is a molecule that's called NAD+. NAD is an empty taxi. So when we break the frame apart, we're going to be carrying off hydrogens on this taxi cab. The empty taxi we call NAD+. When it's full, we call it NADH. And that's what they're trying to sort of show here. Anytime you sort of see this NADH here, or NADH up here, they are high energy compounds that have gotten their hydrogens from either stripping them off of a sugar molecule, or can we strip hydrogens off of other organic molecules? Yes. Because that's where the energy is, baby. It's in those hydrogens. And fats have twice the amount of hydrogen pound for pound than sugars do. That's why fats have twice the caloric value as carbohydrates, more intense, because they basically have twice the amount of these energized hydrogens. Now we'll see later, I might as well mention right now, when we talk about calories, calories is a unit of heat energy. That's really what a calorie is, the measurement of heat energy.
What happens when glucose is low?
I'm sorry, what?
So what happens when glucose is low?
Is low? That's a great question. Because if your brain and spinal cord especially need glucose to run on. Other tissues can run on other types of fuels, for example, lipids. For example, can be used as a source of energy. We'll see about how that process works. The process of using lipids is primarily a process they're going to call beta oxidation. The reason they call it beta oxidation is that remember that fatty acids I'll just use this right now. So this is sort of showing some fatty acids here. Fatty acids are basically just long chains of carbon and hydrogen. Long chains of carbon and hydrogen. Are all these hydrogens energized people?
Yes.
Absolutely, all right? They came ultimately from the process of photosynthesis through sets of reactions, energized electrons from them here. So we can extract energy from these by a process called beta oxidation, which basically involves chopping off 2 carbon fragments from the end. Almost all fatty acids have an even number of carbons. You might remember that from 109 days. Almost all of them have an even number of carbons. And it makes it easy to oxidize in this way. We're just going to break off 2 fatty acids at a time. That's why they call it beta oxidation, because beta is the second letter in the Greek alphabet, the first letter being alpha. They do the whole alpha, beta, gamma, delta. We're not going to go through the whole epsilon sign. But so when energy levels are low, other tissues can run on fatty acids. But let's get a little ahead of our time. If you completely run out of, I'll do a different one here. It's not on this slide here. When your blood sugar levels are low, your liver has the ability to make brand spanking new glucose. You might have done this in brand spanking by using fragments of other organic molecules, especially a group of amino acids. There's one group of 8 amino acids that are glucogenic that can be used for making new glucose. So your liver can make that, and the process is called gluconeogenesis. Did you cover this process, gluconeogenesis? Some yes, some no. Some people are just going, huh? So let's take a look at that term, because we'll see that term again. Gluconeogenesis. Something your liver does. Break this term down. Gluco referring to sugar, glucose. Neo, new. And Genesis, beginnings. The book of Genesis is the book of beginnings. So gluconeogenesis translates as sugar new beginnings. Something your liver can do. Your liver can make new sugar. So if you're, for example, fasting for a long period of time, not taking any new glucose, your blood sugar levels would go down. Your body will release a hormone called glucagon. You might have done a 109. Remember glucagon? And glucagon's job is to try to get your blood sugar levels back up. First, if you have any leftover glucose stored as glycogen, remember glycogen is a glucose storage form in your liver. It will convert it it'll start breaking out glucose. They call that Glycogen Olysis, the breakdown of glycogen. But if you run out of glycogen, you don't have any more, then your liver can make brand new glucose by the process of gluconeogenesis. But in order to do this, it generally needs other scraps, especially amino acids. As I mentioned, there's like a group of 8 amino acids that can be used, that are glucogenic, that can be used for this process. But you don't have much in the way of just free-floating amino acids. The overwhelming majority of your amino acids are tied up in what? In proteins. And most of your proteins are in your... Most of your proteins are where?
Muscles?
In your... Yes. It took you that long to figure that one out from... I guess I need to work on my posing, I guess, here. Right. So the problem is... That's so wrong. The problem is then, in order to make new glucose, your body's going to start to waste away your muscles. And that's the price you're going to pay for gluconeogenesis, is that your lean body mass is going to start to go down. That's the issue with that. I don't know if I answered your question or not, or if you're falling asleep on me here, so I guess I'm boring you with that answer, or it's just not what you intended, or...
No, I'm not as asking.
Okay. So we'll see this process again a little bit later here, but back to our story here. So when we are trying to get energy from glucose, all right, the first step we said was called what? Glycolysis. And it ended up producing these 3 carbon sugars that were called pyruvate. Pyruvate is really a 3 carbon sugar. If it's a 3 carbon sugar, how many hydrogens would it have, each pyruvate?
3 carbon sugar. 36.
36.
Wait, what? 3 carbon sugar.
A 3 carbon sugar would have how many?
6?
6. You always have that ratio, right? Twice as much hydrogen as you have carbon and oxygen. Right? So the 3 carbon sugars would have 6 hydrogens with them. Just pretend you remember this. Yes.
Yes.
So what do we do with this now? What happens next? What happens next will depend on whether or not we have oxygen present. If there's oxygen present, we can take these products into the mitochondria, which is what they're sort of showing here. We can take this in the mitochondria and finish stripping it down, getting more energy out of it, only if there is oxygen present. All right? They will call that process then oxidative phosphorylation. Requires what element?
Oxygen.
We're going to see it's going to be the final electron acceptor. But what if there's no oxygen present? If there's no oxygen present, here's the problem, right? You can't keep running glycolysis non-stop if there's no oxygen for one reason, is that in order for this process to occur, you need a taxi cab. You need empty taxis that are shuttling off what element? The hydrogen. So, and if there's oxygen present, we can take these NADHs and dump the hydrogens on this electron transport system and then just renew them this way here. That's the normal way. If there's oxygen present, we take the NADHs into the mitochondria, they drop off their passenger, the hydrogen and their electrons, and they go back for more. They just keep doing that back and forth. But what if there's no oxygen present? We can't dump the taxis. Eventually, we run out of empty taxi cabs. And then glycolysis would do what? Come to a standstill. But different organisms have ways around this. If there's no oxygen, I'll write that over here. We can dump the hydrogens back on pyruvate. We just dump the hydrogens back on pyruvate. That's what animals do. And as we dump the hydrogens back on pyruvate, we end up with a compound that's called lactic acid. And they'll call this process lactic acid fermentation. Now, fermentation is an umbrella term for all sorts of anaerobic. It's anaerobic because there's no oxygen present. A lot of anaerobic processes that allow glycolysis to continue. And a lot of different organisms use a lot of different forms of fermentation. They call this form that animals use lactic acid fermentation. And you know what the lactic acid buildup feels like, right? Everybody have understanding of what that buildup feels like. Especially if you've been like painting your walls or your ceiling, you've been reaching up and your muscles get sore. Oh my God, I just can't keep going. That's that lactic acid. All forms of fermentation have end products which are slightly toxic. Lactic acid being an example of one. I won't test on what I'm about to say, but other organisms like yeast will take the pyruvate, they'll remove a carbon dioxide from it, plus the hydrogens, And that produces a 2-carbon fragment that we call ethanol. And they call this process alcoholic fermentation. Again, the end product slightly toxic, but can be so nice in limited quantities. Just imagine how different your life would be if instead of your body utilizing lactic acid fermentation, that your body utilized alcoholic fermentation. Would that change your life? It would change my life, all right? Saturday night, getting ready to go out, you just paint your walls, man, all right? You get a nice little buzz on. All right, just me.
Teaching this time.
Okay. I'm corrupting my class here. Okay. So, we've got our pyruvate here. What do we do with it next? Well, if there is oxygen present, we're going to ultimately modify it and take it into mitochondria. We can finish processing it. We convert pyruvate, we're going to do this, what they call a transition reaction. I'll take my pyruvate, which had how many carbons? 3. I'm going to remove a carbon, and carbon always comes and goes in the form of carbon dioxide, CO2. That's how it came during photosynthesis for the plants. That's how it's going to leave in animals during this process here. It is exciting, yes, I agree. All right. In that process, we're going to attach a little carrier molecule that's just called CoA. And we have our end product, an important end product that's called acetyl, ACET, acetyl-CoA. That is closely related to the acetylcholine that you're familiar with as a neurotransmitter. Now, somebody help me here, because I'm breaking my pyruvate from a 3-carbon down into acetyl-CoA that would have how many carbons? 2. How do we come up with 2? Because we lost one in the form of the carbon dioxide. Question. Are we breaking the frame apart when we do this?
Yes.
So what else has to be done?
Hydrogen.
We also have to shuttle off hydrogen. And the process also will produce NADH. So now I have these high energy compounds. I've got acetyl-CoA, I've got NADH, which is a high energy compound that we got from this transition reaction. We have NADHs that we got from the process of glycolysis. All these high energy hydrogens, but we still have a lot of chopping down to do here. Because this acetyl-CoA would have how many hydrogens? 4, right? How did you come up with 4?
The ratio of
Always that ratio, right? For every carbon, there's always going to be 2 hydrogens. Now we take that into the mitochondria, and we're going to process it in what they call the citric acid cycle. Let me explain this cycle to you.
It's a lot.
The citric acid cycle is a chop shop. Do people know what a chop shop is? If you're living in Philadelphia, you should know what a chop shop is. That means that if your 2010 Toyota Corolla gets stolen, is it likely that somebody out there is actually then driving around in your 2010 Toyota Corolla? No. What did they do with your 2010 Corolla?
They take it apart and sell parts.
They take it apart. That's what a chop shop. They take it because the parts are way more valuable. That's what a chop shop is. And the citric acid cycle is a chop shop. Let me show you the basics of this chop shop here. I'm going to give you about 5 more seconds to memorize this entire slide.
Got it.
Okay, good. Let me explain what's happening here. Let's see if you can explain it to me here. All right. So here's how this chop shop works. All right. I'm going to start you off, and you tell me how it ends. All right. I'm going to tell you the first page of the book. You're going to tell me what the ending is. So, first page of the book. We're going to take this 2-carbon sugar and attach it to a 4-carbon sugar, making it a 6-carbon sugar. And then somehow we're going to chop shop it apart until we get back to the 4-carbon sugar that we started with. We're going to take our 2-carbon and attach it to a what? 4-carbon. And then we're going to chop that 6-carbon sugar down until we get back to our starting 4-carbon sugar. So we're starting to attach our what? Our 2-carbon sugar to our what? 1, 2, 3, 4-carbon sugar to end up with what? 6-carbon. Okay. But in order to get us back to our starting place here, right? All right. The acetyl-CoA that we brought in had how many carbons? 2. And how many hydrogens? 4. Which means to chop it down, we need to account for how many carbons? 2. And how many hydrogens? 4. So notice that the carbons are going to go in the form of what? CO2. You see it getting rid of carbon dioxide here and here? Right? And how are the hydrogens going to be removed? Do we have a taxi that can carry them?
NAD.
We have our taxi, which is what? NAD. So now there are 4 hydrogens here. We have to account for. But breaking the frame apart is going to liberate a little bit of ATP directly, but that's going to de-energize one of the electrons. One of its electrons is going to be de-energized a little bit so we can get a molecule of ATP. Here's my ATP that's going to be made directly, which means that there's going to be one of those 4 electrons that we have to carry that do not have quite the same amount of energy. 3 of them are going to be carried out by our high energy carrier that was called NADH. All right? One of them has to be carried on by a second rate carrier that's called FADH. Where's my FADH? It should be over here. So all FADH is like, one of the, I don't know. It's just not quite as good, not quite as energy as you have. It's like I'm trying to think of, it's like being like one of the lesser Kardashians, not one of the top sort of ones, Is that wrong of me to say it like that? Probably wrong.
Also, she's like the Kendall. She's like the Kendall.
Oh, okay. So, or just think about the NADH as being like an Alex Baldwin, and then like the FADH as being like some of the other like less talented Baldwin brothers.
Oh, yeah.
Who I don't know how they end up with careers, and I'm stuck here, but that's not the issue, is it really here? No. So why am I bringing this all up right now? Because we finished stripping it down. That was the whole idea behind the citric acid cycle, right? Is that we've chopped it down into its pieces, the citric acid cycle. And the citric acid cycle has produced lots of NADH. For every turn around it, we get 3 NADHs, but we also get 1 FADH. It's sort of a lesser quantity, you know, like a Billy Baldwin, you know, or... Okay, so what do we do with them now here? All right, well, here's the beauty that now we get to harness all the energy from all the NADHs from every source that we got, all the sources, from glycolysis and the transition reactions. We're going to take them all to this electron transport system. And here's where the big payoff is. Let me explain how electron transport works. So this process occurring in the cytosol or the mitochondria? Everybody hopefully was going to say mitochondria, it's all mitochondria. So here's what we're going to do. We're going to take the energy from the electrons that are accompanying these. We're going to use their energy to pump hydrogens from one side of the membrane into an enclosed space. Now, here's the beauty. Once we pump hydrogens in here, now we have a high concentration of hydrogens, right? We've been using energy and electrons to do that, right? Are these hydrogens going to want to flow out? If we've been pumping them in, are they going to want to flow out?
Yes.
Right, that's diffusion, right? And we let them go through a magic molecule that's called ATP synthase. Write this shit down. ATP synthase is driven by the flow of hydrogen ions. ATP synthase is driven by what? The flow of hydrogens. On a one-to-one basis. What did I mean when I said a one-to-one basis? What that means is that for every 1 hydrogen that flows through it, it generates enough energy to regenerate 1 ATP. I like to say we regenerate ATP instead of saying making ATP. That's my own personal preference. Because we're not making it from scratch. All we're doing is regenerating it from the broken down pieces, which were the ADP and just a phosphate that's floating around inside the cell. That's what that little I means. It's an intracellular phosphate that just happens to be handy. Now, NADH has enough energy in its electron to go to 3 carriers, boom, 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 which means it can pump 3 hydrogens. If NADH can pump 3 hydrogens, then how many ATPs can it regenerate?
3.
3. So every NADH is going to be worth what? 3 ATPs. FADH, or Billy Baldwin, only has enough energy to do it twice. All right. So it's going to come in a little bit later. So FADH can only pump 2 hydrogens, which means it's only going to be worth 2 ATPs. But the overwhelming majority, think about this, the overwhelming majority of all the hydrogens are being brought over by what molecule? NADH. Did we get NADHs in glycolysis? Everybody should say
Yes.
In transition reaction?
Yes.
Yes. In the citric acid cycle?
Yes.
So ultimately, since the overwhelming majority is going to be carried by this, how many ATPs can we get from one sugar molecule? Well, 12 hydrogens are almost all carried by NADH. And if each NADH is worth how many?
3.
3. Which is like each glucose molecule has the exchange value, like we're exchanging dollars for euros, has basically an exchange value of roughly about 36 ATPs. This is why cells like to actually store more of their energy as glucose, all right? You can't store a lot of energy as ATP, it's just too big and bulky, all right? But one sugar molecule has enough energy to regenerate what? 36 ATPs. And you just do this on the fly as your cells need energy. This process is basically going on a demand basis. When the cells need it, when ATP level starts to run low, let's start to produce more of it using these mechanisms here. Now the process of breaking down fatty acids we call beta oxidation, because we're breaking off what, 2 carbon fragments at a time? 2 carbons is probably worth how many hydrogens?
4.
4. Which means that each beta oxidation we're going to get 4 NADHs roughly out of it. Means we're going to get a lot of ATP for that process. Now, proteins, if you're starving, can be used for energy too. Or if you're on like one of these, paleo diet things where you can only eat what you have killed with your own hands or whatever they're doing now with those paleo kind of diets here. The issue with this is that you're going to break it down into amino acids, all right? And almost everything gets broken down by this citric acid cycle, this is a key thing here, by converting it into acetyl-CoA. The fatty acid, the 2 carbon fragments get converted into acetyl-CoA. Amino acids, most of them get converted into acetyl-CoA. It is a common entry point for utilization of almost every kind of fuel. Now, there's a bit of a lie in there. Can't stop myself, it's pathology. There's a few amino acids that actually enter the citric acid cycle at a different point, not at the acetyl-CoA, but those are a rare number of them that I'm not going to hold you accountable for. That's really for a full semester course. I'll be happy if you know that most things are going to enter via acetyl-CoA. The breakdown of carbohydrates does it through acetyl-CoA. The breakdown of fatty acids does it through acetyl-CoA. The breakdown energy of most amino acids through acetyl-CoA. Now there is an issue here with breaking down amino acids, is that amino acids contain that nitrogen group. It's called an amino acid because one side of it has this amine group on it, right? C double bond O. Which here's my very rapid view of a amino acid. They look nothing like this, but that's my best guess. So in order to actually process this, I'm going to need to strip off the nitrogen as a waste product. Now, different forms of nitrogen waste. Let me just mention this real quickly. The easiest way that organisms can get rid of nitrogen is just add a hydrogen to it and you have NH3. I'll just write that in, NH3. And they call that compound ammonia. Ammonia. Which is a little bit toxic. Now, fish, as if anybody cares, but I will share with you about fish. All right, fish are surrounded by, what's the stuff they're surrounded by?
Water.
Water. So they don't have to worry about ammonia accumulating this because it just is going to go into the water. So fish will get rid of their nitrogen waste as pure and simple ammonia. And by the way, this is one way you can sort of tell if you're shopping for fish at the market, if it's fresh or not, because if it starts to get a little bit old, it's going to start to smell like what? Ammonia, because of this breakdown process that's going on. So give your fish a quick little sniff while you're there in the market there to make sure it doesn't smell like what? Ammonia. For us though, for animals though, this is too toxic to build up. So what we do is we're gonna actually attach two of them to basically a carbon dioxide on either side. You don't need to write this down, but we call that compound urea. Urea is simply a less toxic form of nitrogen waste. Urea is a less toxic form of what? Nitrogen waste. The nitrogen from the catabolism of amino acids. And how will you get rid of the urea?
Urine.
Urinary system will get rid of that. Now we'll see this term again later, but this term called transanimation. Transamination is that this process of transamination is a process of converting one amino acid into another by simply moving around the amino group from one onto another one. So your body can convert many amino acids into other amino acids by the process of what? Transamination. Now, having said that, something we'll come across a little bit later, just to sort of set you up, is not every amino acid can be converted into every other amino acid. There are some amino acids that you cannot make from other ones. You have to get from your diet that are considered essential amino acids. So there are some amino acids that are essential amino acids, others that you can make on your own. I only bring this up is because to get the right assortment of essential amino acids, if you have a diet that has, if you are a vegan or a vegetarian, you have to be careful about getting your proteins to make sure you're getting the right amount of all your essential amino acids. You get the right balance of legume protein and nut protein and things like this. But if you have a diet that has some dairy, has some meat, you don't have to worry about getting the right amount of essential amino acids. You're always going to have them. It's only people who are vegan, who can certainly get the right amino acids. You just have to be thoughtful in your diet to make sure you're mixing the right amount of legumes. Soy has a lot of great ones in it, which is why soy is such a common component, staple of most vegan diets here. Okay, so enough about this. Let's talk about eating. Because as we've sort of seen, that the only thing your digestive tract can really absorb are the monomers. Everything has to be digested by the process of hydrolysis. Would hydrolysis be catabolic or anabolic?
Catabolic.
Would be a catabolic reaction, right? Is that going to release some energy just digesting food? In fact, you generate heat while you're digesting food in that process of breaking them down so that you can sort of absorb them here. And we refer to this state right after you've eaten a meal as being the absorptive state. The absorptive state is the state right after you've had a meal when generally nutrient levels are going to be high, especially carbohydrate levels are high. And this absorptive state will last a couple of hours, 3-4 hours. Well, nutrient levels are going to be high. Now, if you didn't get this in 109, you're gonna get this now. And that the absorptive state is primarily governed by insulin. Insulin release is a hallmark of the absorptive state. Insulin allows glucose uptake, without which the cells are basically starving, have to rely on fatty acids if there's no insulin present. And so, in this case here, glucose levels, and again, glucose is a major source of energy. For you, it's like money. Now, tell me what would happen, for example, if you were to get, what would you do if you got a big windfall of money? What would you do with that money? Big windfall of money.
I want to spend
You
it.
hit the number, people, or your grandmother finally died. God. Did I say that out loud? No, that was so wrong, so wrong. All right. What would you do with that load of money?
I would save it.
Hmm?
I'm gonna spend it.
You're gonna spend it all?
Save it, save it.
You wanna invest?
Are you gonna spend it or are you gonna save it?
Save it.
Yeah.
You're gonna do both?
I wanna spend it.
All right, you're gonna spend some, but you're also gonna
Save
Save
some.
save some. And hello, that's what your cells do, all right? When you get this big windfall of energy, all this sugar, you're gonna do what? Spend some and
Save
save
some.
some. So during this absorptive phase here, you get a lot of ATP generation. You get a lot of oxidation of glucose because you got all this energy. We're going to spend it. Woo! All this energy. We're going to make a lot of ATP. So ATP production goes up. That's the spending. But how are we going to save it?
You store it somewhere.
Well, we start by saving as much as we can in the form of glycogen. Remember, glycogen is a glucose storage form, and they call that process glycogenolysis, or glycogenesis. My bad. Glycogenesis. Glycogenesis.
Glycogen.
Glyco for glycogen. Genesis means beginnings. So glycogenesis is the process of making what? New glycogen. Remember, glycogen is just a polymer of glucose. So we're going to start to store a lot of the glucose as glycogen, pretty much in two places. Your skeletal muscles will store it for their own selfish, personal use. Selfish muscles. I want it all for myself. No, you're not getting any of it. That's what skeletal muscles sound like. So they talk about, for example, athletes who are carb loading. Have you heard that term before? Mm-hmm. What they're trying to do is to get their muscles to store the maximum amount of what?
Glucose.
Glucose in what form?
Glycogen.
Glycogen. Now your liver also stores it, but your liver stores it not for selfish use, but for body-wide distribution if your blood sugar levels start to get low. But even then, there's only a maximum amount that your liver can store as glycogen. As a rule of thumb, don't write this down, but just listen. As a rule of thumb, animals do not like to store much of their long-term energy as carbohydrate, as glycogen. They prefer long-term energy stored as fats, lipids. Why?
Because stores longer.
here's the reason behind it. Carbohydrates are extremely hydrophilic. They're water loving. So much water loving. It's not just they dissolve in water, like glucose and other small things dissolve well in water, but large polymers like this will suck water with them. For every 1 gram of carbohydrate that you store, 3 grams of water come along with it. That's how hydrophilic it is. Do you have that issue with long-term lipid storage, fat storage?
No.
Are you going to have that same water weight gain with
No.
lipid storage? No, because lipids are hydrophobic. That's why animals don't like to store a lot of their energy as carb. A lot of plants will, like a potato. It's like pure carb, right? Rice, grains like this. Why can plants get away with it? Why don't plants have to worry about that? Because they're not, thank you. It's really that simple. They are not moving around, all right? They don't have to lug all that water around with them, do they? Are you yonder? If you like see broccoli like walking down the street or something, people, you call one of us, okay? And we'll make sure that you get taken care of, all right? Shouldn't be happening. That's why plants can get away with it. In fact, for plants like potato, they can actually use it as water storage. In case there's a drought, there's a dry spell or something like this, they can get away with that. But the reason I'm bringing this up is that there's only a limited amount of glycogen that your liver will store. After you kind of reach the maximum, it will start to convert excess glucose into fat, into fatty acids. And that process they call lipogenesis.
Is that what a fatty liver is?
Hmm?
Is that when they say fatty liver?
Fatty liver, because the liver is working overtime to do this, yes.
Converting to fat.
Converting sugars into fats. And what hormone was involved with this?
Insulin.
Insulin. So if you're carrying a little extra body fat, who can you now blame? Or what hormone can you blame?
Insulin.
Insulin. You do that to me, insulin. Bad insulin. Has nothing to do with my dietary choices. No, it's just stupid insulin. It's making me fat. So we have here again, during the storage of excess, again, primarily as adipose and adipose tissue. This is the absorptive state here. Promotes, again, lipogenesis in adipose tissue and liver. All right. Amino acids can be used, again, for protein synthesis. Big deal here. Growth hormone, don't worry about here. That's an aside here. And this is a nice little slide that's sort of showing what's happening during this absorptive state here. They're showing this would be your small intestine someplace, probably the jejunum, where almost all the absorption has taken place, because you all remember that. That we're pumping in the glucose, amino acids, fatty acids, glycerols in your bloodstream here, and in response to the pancreas secreting the hormone insulin, because your blood sugars are high, the cells will take up the glucose, store some of it as glycogen, others will be converting to fats in the liver. Bad liver for doing that to me. And again, adipocytes also taking up some of the excess lipids directly. So, we finished your absorptive state. It's been like half a day since you've had a meal. 3-4 hours. What's happening to your blood sugar levels now? They are going down because of insulin. And now we have reached the post-absorptive state that will last now until your next meal. if that next meal is 2 or 3 days away, you're going to be in that post-absorptive state for 2 to 3 days until your next meal. And the hormone that governs the post-absorptive state is glucagon. Glucagon is the major hormone that mediates the post-absorptive state.
Do we have to know those numbers? The blood glucose concentration?
Oh yeah, I'm gonna make you memorize every single freaking number on here, yes.
Well,
No.
I don't know, sometimes you put numbers on the exam, you know, so.
I did, this won't be one of them here.
Okay.
And the other reason why, it's a cheat for people who have diabetes, because people have diabetes, they know this number like the back of their hand, all right? They know that they're looking for a number around 100 parts per deciliter, all right? They've been knowing that for their entire diabetic life here. And so when people are monitoring their blood sugar levels, this is what that's what they're looking at here, trying to make sure it stays within that range. So again, the hormone that's really mediating this phase was which hormone?
Glucagon.
Here's the thing. Unfortunately, we have two things, glycogen and glucagon, which are two very different compounds that have unfortunate similar names. Glycogen is the way that we store sugar. Glucagon is a hormone that helps to regulate blood sugar levels. It's unfortunate that the names are that similar because it does tend to lend to confusion. But the key thing about glucagon, it really has just one target, and that's your liver. When your blood sugar levels are low, your pancreas will start to secrete glucagon. And glucagon really only has one target, that is the liver. Now, what can the liver do in response to glucagon if your blood sugar levels are low? Well, if your liver has stored glucose as what form? Glycogen. It will start to catabolize the catabolism, the breakdown of the glycogen, to release the glucose monomers into the bloodstream. And they call that process Glycogenolysis. Does that name make sense?
Yeah.
Break it down for me. Glycogenolysis. Glycogen is glycogen, and the lysis means what? To break, to split. So glycogenolysis is the process of breaking down glycogen to release the sugar molecules into the bloodstream. But what if there's no more glycogen left? What if you run out of all your storage form of sugar? Is there still a way to get my blood sugar levels up if I run out of sugar? Can I make brand new sugar?
Yes.
Can the liver do that?
Yes.
Right. Gluconeogenesis. That's that process of gluconeogenesis again. So that hormone glucagon will stimulate that process of gluconeogenesis. And of course, what are you going to be losing? You're going to be losing some lean tissue, lean protein tissue, because that's what
Your muscle?
that's what you have to run on. Now, while the liver is doing this, other tissues are trying to save the glucose for the organs that absolutely have to have it. Do you remember what organs have to run on glucose? Brain, spinal cord, neural tissue has to run on glucose. Can other tissues run on alternate fuels like fatty acids?
Yes.
Using that beta oxidation, right. So most tissues, if your blood sugar levels start to run low, will start to rely more on lipids for fatty acids as their source of energy, saving the glucose for brain, so the spinal cord, because they'll do that. So we have glucose conservation, so we're going to use fatty acids. So other tissues, your muscles can use this, your heart can utilize them, and even ketone bodies here. Other alternative sources here and here. And again, the big thing here, the big hormone was glucagon. I'm not going to ask about other things that are on here because they're not going to be as important. You might have read about glucocorticoids. Did you come across glucocorticoids in 109? Okay, so you remember that, why do I say things like that? The adrenal glands that sit on top of your kidneys, your adrenal glands here, have an outer cortex, the adrenal cortex, and an inner medulla. The adrenal medulla, the inner part of the adrenal, was the part that secreted epi and norepi when you're in fight or flight mode. Adrenal medulla was an extension of your sympathetic nervous system. The adrenal cortex produces steroid-based stress hormones, like aldosterone that helps you to conserve water and salt. And the main one that it produces is called cortisone, or cortisol, that helps you to conserve sugar. Because when you're long-term stressed, your body doesn't know that there's a candy bar in your purse or in the corner. It's like, let's conserve sugar just to make sure your brain and spinal cord have enough of it. So, because hormones like cortisone that come from the adrenal cortex are a steroid, often they will lump those together and refer to those hormones as glucocorticoids. Glucocorticoids, the prominent one is cortisol. Gluco, because they affect sugar. And the cortico is not from the cerebral cortex, it's from the adrenal cortex. So glucocorticoids can help to stimulate lipolysis, which means the breakdown of the triglycerides and release the fatty acids. You're not going to be tested on this little bit, but I'll still share it with you. If people have high levels of these glucocorticoids because they're taking an inflammation or they've got a tumor that's produce too much of them, right? They will start to undergo lipolysis, they'll break down their lipids, but if you are not burning the lipids, they get redeposited in different locations. So a common form of having too much of glucagon is something like called Cushing syndrome. People might have heard of this like that. And you end up, for example, with a lot of extra fat deposits around the face and the back. You get what they call a buffalo hump here because it's redepositing all the lipids that were released by this process. Okay.
Very cool.
And this is just sort of showing what's going on in that post-absorptive state here, is that the pancreas is releasing what hormone in the post-absorptive state? Glucagon, right? The absorptive state, what hormone is the absorptive state?
Insulin.
Insulin. Insulin is involved with what state?
Absorptive.
Glucagon is involved with
Post.
post-absorptive state. What hormone is involved in the absorptive state?
Insulin.
What hormone is post-absorptive?
Glucagon.
What is insulin involved with?
Absorptive.
Absorptive state. Glucagon.
Post-absorptive.
Post-absorptive, good. All right. You just have to hammer this over and over and over again. I know it's annoying, but it's the only way to get it through sometimes here. So your pancreas is secreting what hormone in the post-absorptive state?
Glucagon.
Glucagon. And what's its main target?
The liver.
The liver, triggering the liver to release the sugar that it has already stored by the process of glycogenolysis. But if it's run out of glycogen, it will now need to make brand new one by the process of gluconeogenesis. And that's why they're showing this poor skeletal muscle up here. Poor skeletal muscle, we're gonna have to break it down to release what? The amino acids that we can use to make brand new sugar. I think that's probably enough for today,
Yes.
since it's already 2:31.